All right, so earlier you studied mitosis. Okay, so mitosis is where one cell divides into two, and now we're going to look at the process of meiosis. Right? And so meiosis and mitosis are similar in certain ways, but very different in others. Starting off with their purpose, right? And so the the purpose of this particular lecture is to give you not all the details, but some of the major differences between mitosis and meiosis, and then what are some of the unique events and the terminology, the terms associated with the process of meiosis. Okay, so that's kind of our point here. So what happens in mitosis? All right, we have one cell, goes into two cells. Those two cells should be both identical. So I mean, they should be identical to each other, and they should be identical to the original cell, right? So that's the idea here. So it's about cell growth. One cell divides and you have two. They're the same. Those two cells divide. You have four. They're the same. Those four cells divide, right? You keep going and keep going and keep going, doubling the number of cells. And all those cells should be the same unless there's mutation or errors that cause the cells to become different, but they should not genetically be different from one another. Meiosis is different. And now we're looking at meiosis. What we're going to have is one cell will become two cells, but then it's going to divide again into four cells. So we're going to end up with four cells in the end. It's kind of like what we're looking at over here. These cells are going to be unique, not only from one another, but from the original. So that you're going to now be creating uniqueness. So that's kind of the point here of meiosis, is to create something genetically unique or different. Whereas mitosis is not. Mitosis is just to double the total cell number. So growth. So if an organism has a few cells and it's getting bigger and it needs more cells, the cells are dividing. Right? Your skin cells are constantly dividing and creating new skin cells that replace old skin cells that die and then fall off, and then new skin cells replace them. They should be the same. They should not have mutated, but they can mutate and become different, but they shouldn't. They're not supposed to do that. That's through mitosis, making them the same. In meiosis, this is unique. So what's the purpose of meiosis? So again, why do this? It's typically for reproduction. And get some genetic diversity, or to increase genetic diversity. So this is going to be a theme now throughout the rest of the course, right? Because what we're going to be focusing on, I'll take big picture from this point forward in the course, we're looking at the process of meiosis, which the reason we're doing that is to lead into genetics and inheritance. As we go over some basic patterns of inheritance, we'll then apply that to populations of organisms to see how species get genetic diversity within a population, which is then through selection, so events, stressful events, select for certain characteristics or traits. And then those organisms are successful and survive, whereas others may die off or not be able to reproduce. And over time, potentially, there could be change, right? Which means evolution of some sort. That would then lead to biodiversity, diversity among organisms. And so that's kind of like the whole rest of the semester of this course, right, is looking at diversity of organisms from fungi and plants and animals within those groups, the diversity within the groups. And what we are looking at now is the explanation, a little bit at least, of the explanation of how. How does genetic diversity occur? Where does it even come from? How is any of this possible? So we're going to be looking at it at the cell molecular chromosomal level of change and diversity. So that's kind of the point of this, plus the next two things, which we'll get with Mendelian genetics and population genetics. Right? And so they all will connect. And then the overarching connection to the whole remainder of the course is that explains relationships between organisms, how they can be similar and then different, and where the genetic basis lies for that. So going back to the process, what we're supposed to focus on now is meiosis. Right? So what actually happens in meiosis, and we're going to talk about the terms, and then we'll go over, like I said, not all the details, but some of the details of the process. So what's going to happen first is you're going to have a cell, right? And this cell is a germline cell. What does that mean? Well, and this is a little bit different. We're going to get into later in the course. For organisms that reproduce sexually in the way that you're familiar with, say there's a sperm and an egg and there's fertilization, like that, right? Only certain cells can create the sperm or egg, not any cell in the body. So in the human body, a skin cell can't produce, sperm or egg, right? Only certain germline cells, which are the cells that will produce the gametes. Other cells of the body are somatic cells, and they can only undergo mitosis. So most all cells can only do mitosis, they can't do meiosis. Only a certain group of cells can undergo the meiosis process. So mitosis is very broad. Everything kind of undergoes mitosis. Meiosis is unique. Not everything can do this process. Only a certain group, subgroup of cells can do this process. We'll find in some organisms, which is where it gets kind of like weird, right? Their body cells are haploid or have half the number of chromosomes like the gametes, and that cells of the body of one organism will actually fuse with the cells of the body of another organism in a fertilization type process that happens in fungi. So it's very weird, very different, but conceptually, the idea is going to be the same. There's going to be a meiosis process in fungi. It will create haploid cells with half the number of chromosomes. When it occurs, there's going to be a lot of genetic diversity created. And when that fusion occurs, which is different, it's called plasmogamy, which is what we'll get to that later when we talk about fungi. It's like fertilization. Then it's going to be mixing of genetic information to create a unique new individual. And then the process will continue. So that's kind of the idea here. We're starting off with cells that are what we call diploid. So di meaning two. That means two of each chromosome. So in this particular model, all right, what I'm going to do is say that there's another term here we'll talk about here called karyotyping. Now, organisms, typically as a species, partly get defined by the number of chromosomes they have. So some organisms are different species and they have the same number of chromosomes, but there are often times where different species of organisms will have different numbers of chromosomes, just different, not just genes and specific information, but just even the total number, like how many do they have in their cell. There isn't a set number for living things. It varies among those different living things. There's an example of that I read where a breeding program at a zoo was trying to help out an endangered species, and they were failing, and they were unable to produce viable offspring, right, from the animals they were trying to mate. And what they ended up finding out is by looking at the karyotyping, some of the individuals they were using had different numbers of chromosomes. There were these deer. They were actually different species of deer, but they looked so much alike. They thought they were the same, and that's why they were failing in their program. So karyotype, so that word karyo, right, is referring to, right, the nucleus and the chromosomes. Okay, so how many chromosomes does the organism have? Okay, that's how many different chromosomes. So in my little model here, I'm coding them, right, with some color and size. Okay, so what we have here is is the size is for the karyotype. So sorry, these little ones are chromosome, say number 1. Okay, and these bigger ones are going to be chromosome number 2. All right, so you see these two are longer and these two are shorter. So these two short ones are the same, chromosome number 1. These two are a different chromosome. So we could have many of these in here, and the karyotyping would be looking at physical characteristics, length, and shape, and a variety of things to actually find out like how many different ones there are. So what do I have the different colors for? Well, that's because you're going to notice here, well, we have 2 of chromosome number 1, like the blue one and this pink one I drew, right? And then 2 of chromosome number 2, the blue one and the pink one here. Why? Well, because one of them came from one parent and the other came from the other. So let's just say, just for stereotypically keeping it just, well, actually, we'll flip it stereotypically and say that the pink ones are from the father or the paternal, and the blue ones are maternal. Right? So the mother gave chromosome number 1 and chromosome number 2. The father gave chromosome number 1 and chromosome number 2. So you have one of each from each parent. That's why you have a diploid cell. So all of your cells in your whole body are like this. Okay? They're all like this. Now we have, right, 23 different chromosomes in the cell. That'd be too much to draw here, and it would be just too confusing to even use in a model or explanation. So I only have 2. So just keep in mind, most organisms are going to have more than 2. This is just to help explain this a little bit easier. So the first thing that's going to happen is DNA replication. So we'll go back in the class. We covered DNA replication, which you had before mitosis, before cell division. Why does DNA replicate? So the cell can divide. You double the DNA. So what's going to happen is that each of these chromosomes are going to undergo DNA replication and double the amount of material that's there. That's a key thing, because often when you see pictures or images, you often only see these X-looking, okay, whatever kind of squiggly images I have here, but that's only after DNA replication. Right? So what do we have, say in the terms here? So the chromosomes that are the same, so say chromosome number 1 from the mother, chromosome number 1 from the father, these are homologous chromosomes. The now duplicates, the one of each, and this goes for all of them here. Those, remember, we refer to those as sister chromatids. And this should be familiar from the lecture on mitosis. Right? So if you do the mitosis first, then you go into meiosis, it'll help. Right? You kind of understand a lot of the basics and the terminology. So where this becomes different is that in mitosis, what will happen is that all of the chromosomes just kind of get attached to the tubules, which I talked about again in the earlier video, and then it starts to try to pull the sister chromatids apart. But that doesn't happen in meiosis until later. So earlier on in meiosis, what happens is meiosis. So this is things that are unique. For meiosis, we have a pairing of homologous chromosomes. So that means the 2 chromosomes that are, so now I'm going to kind of bring this over here before we get to the next stage. I'm going to show you kind of what happens, like how do we get to this next stage. Okay, so partly what happened here was we had these make sure the bigger ones look bigger. Okay, so maternal and paternal chromosomes, homologous chromosomes, are pairing up. So here, these are chromosome number 1, this is chromosome number 2, right? The mother's chromosomes, the father's chromosomes. They now pair up during what's called prophase 1. And so same thing like in mitosis, right? There are microtubule organizing centers that send out kinetochore tubules like this. And they go to pull not the individual sister chromatids apart, but they're going to pull apart the pairs, the homologous chromosomes. So homologous chromosomes are going to separate and go into different cells. So that's one way of creating genetic diversity. Now, I could have drawn this, the way I did, or I could have drawn it this way. I'm going to try to do this really fast. So excuse the extra squeakiness because I'm just going to go a lot quicker. Okay. So this is option B, so this is option A. What do I mean by option? Well, in this particular orientation, what's going to happen is when this cell divides, one side's going to get chromosome number 1 from the mother, but number 2 from the father. And this one's going to get number 1 from the father and number 2 from the mother. This one cell, both 1 and 2, will be from the mother. The other one, 1 and 2, will both be from the father. Right? So it's random. It could be either way. We don't know which way it's going to end up. Right? That's one part of creating genetic diversity, right? Is somewhat of the randomness. So that creates some differences, right? So we're going to get, okay, so that means that in the end, we could have different individual haploid cells, ones with, just the paternal chromosomes, some with just the maternal chromosomes, and some with combinations of maternal for one and paternal for the other. So we can have those different possibilities. That's one way. The other way genetic diversity is created during meiosis, right, has to do with what's called crossing over. And this is the whole big thing, the big thing about meiosis. Often called by a lot of different names. Okay, so there's synapsis and genetic recombination. And some of these could be defined as being slightly different than others, but they're all essentially getting at the same idea or the same concept. And that is that during this part of meiosis, when the homologous chromosomes pair up, which is unique, doesn't happen in mitosis, only happens in meiosis, that these guys, based on the way that they're oriented to one another, a little bit of this chromosome and a little bit of this chromosome might be touching each other, you see, or crossing over like this. Now, when that happens, there can be an enzyme. There is an enzyme, right, that can splice the DNA. So essentially, it will be cutting off a piece of one chromosome and then reattaching a piece of the other chromosome to it. So that means that in the end, what's going to occur is that this maternal chromosome is going to get a little bit, a few genes from the paternal, from the father's chromosome, are going to be attached to a mother's chromosome. And to this chromosome that came from your father, well, a little bit of a chromosome from your mother is going to be attached to it and create this sort of hybrid chromosome, right? And that can happen to both of them. I drew them both happening here at the top end. They can happen anywhere. They can happen more than once. You can have multiple crossing overs. Okay, you can have a whole bunch of them occurring. How many occur? Where do they occur? That is random. And every single time the process occurs, it can happen in different places and it can happen a different number of times. So what that means is that this can lead to potentially infinite genetic diversity because it's completely random how much is going to cross over and where those crossing overs will be. So what we can end up with in the end are chromosomes in cells that are different from each other. And every time the process occurs, you could end up with different ones that are different from the previous time that it happened. So diversity, 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 new unique cells. So this is just kind of the most simplistic summary I can give you with an example of just having the one occur between these, like this example with two chromosomes. But again, as there are more chromosomes, see, some of them may not have it occur during them at one point in time. And then another time they have it occur multiple times. Other chromosomes switch. So one chromosome, a different chromosome number 4 has crossing overs, and chromosome number 5 doesn't. And the next time chromosome number 5 does, and 4, and or they can all have it. All these things are possibilities, which means there's so many different possibilities. We can create a lot of diversity in these final cells that we make. So what's going to happen then is when meiosis begins, the DNA will double. The first thing that's going to happen. Then the homologous chromosomes pair up. Right? So this is kind of like one of the key steps. Pairing of homologous chromosomes. That's unique for meiosis. Crossing over. That's unique for meiosis. Right? We're going to get some genetic information recombined, different combinations, of genetic information on different chromosomes. Then what will happen is the separation of homologs. So the homologous chromosomes are separated into different cells. So there's going to be one cell division, so what we call meiosis 1. And in meiosis 1, the homologous chromosomes separate. Okay, so these separate, these separate. The sister chromatids didn't separate. The sister chromatids, the doubled DNA, are still together. And remember, in mitosis, these would be identical. Sister chromatids, that's a definition. Identical double-stranded DNA is created by DNA replication. But now they're not identical. Because of crossing over, you have one potentially, of them is hybridized, and it could be in multiple locations that it could be hybridized like this, where it's crossed over. The process is going to repeat, and it's going to repeat in the same sort of way it would have. So let's just say we're going to go with this cell. So this cell was not gone, it's just we're not focusing on it, okay? So now we're just saying on one of these cells. Now we enter prophase 2, second one. Okay, so crossing over has already occurred. What's going to happen here? Basically the same thing that happened in mitosis. So now the second part, so meiosis 2, similar to mitosis. What does that mean? That means it's the sister chromatids that separate. But because this is a second division, now we're not going to have, say, 2 of chromosome number 1. So say, right, this is chromosome number 1, this is chromosome number 2. I should have 2 of chromosome number 1, but I don't. I only have 1, right, of them. I should have 2 of chromosome number 2, but I don't. I only have 1, right? So these are going to be haploid. Look, that's very floppy. Haploid cells. So also written as 1N, meaning they have 1 of each chromosome as opposed to 2. So diploid is 2N, haploid is 1N. So meiosis 1, right, is where we first just separate homologous chromosomes. Meiosis 2 is where we separate the sister chromatids. The crossing over and recombination, all that happens in meiosis 1. Meiosis So that's the unique part of it. Meiosis 2 isn't necessarily that unique or that different than mitosis, except that we end up with different cells in the end, and they're haploid cells. Right? They only have half the number of chromosomes, right? Number one, number two, number one, number two, number one, number two. They're all haploid. You can see they're all different. Let's take a look at the chromosome number twos, right? This one is mostly the maternal chromosome, but it has a little bit of the paternal mixed with it. This one's all maternal, 100%, all right? Didn't get mixed up in any way. This one's all paternal, didn't get crossed over in any way. This one is mostly paternal, but has a couple maternal genes, so they're unique. So each of these cells is genetically different from the other cells. Those cells could then become, through a process of gametogenesis, the gametes. So one of them may become the ova or the unfertilized egg, or they all may become sperm cells, right? Or they could become spores in some certain organisms, and that spore could give rise to a haploid mycelium, which is what we see in fungi. So this little cell might divide through, it's kind of weird, but it'll divide through mitosis into another haploid cell. So then it'll be two haploid, and then more and more and more and more and more, and they'll all be all identical to this one. And that might form the body of an organism. And we do see that in certain plants. So mosses, right, have this, where we have the most of the body. If you look at the little green, little leaf-like structures of a moss, all the cells that make that up are haploid. They're the result of mitosis that makes them multicellular, but the reason they're haploid is because of meiosis, right, occurring. So there are different organisms do things slightly different in some ways, but the actual process, when meiosis occurs in that moss or when meiosis occurs in that fungi, it's the same. Okay, so whether it's going to be the end result is going to be an egg or a sperm or a spore or some other structure, it's still the same process, same steps, same terminology, all the same things happening. Okay. So for non-majors class, if you know this, you really know more than probably a lot of the biology majors, right? So there's a lot of other details that we could go into, but they're not really something we need to go into. This gives you the background to next start to talk about genetics and chromosomal basis of inheritance. And then later when we talk about alleles on different chromosomes and all the possibilities we have, and we can mathematically look at that, you can see there's a sort of a chromosomal basis for those inheritance patterns, that we are inheriting chromosomes, physical pieces of DNA, and that those physical pieces of DNA can be mixed up in an infinite variety of ways that are set both from the process of separation being random and from the mixing of the alleles potentially being random. So genes from one chromosome being swapped to another. So make sure you know all the basic terms and just the key events of the different processes, and also then be able to say, what is the difference, say, between meiosis 1 and meiosis 2, kind of what happens in the first division, just separate the homologous chromosomes. What happens in the second division, you separate the sister chromatids. How is the whole process different from mitosis? Mitosis produces identical cells. 2N, 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 they're all always the same. And there's no combinations or recombinations. There's nothing that changes about the chromosomes. They should be the same. Here, constant change, nonstop, infinite sort of amount of change occurring, a lot of randomness. Okay, so keep those things in mind, and you should be in good shape with your understanding of that process.